In the previous session, we have discussed about ketose-aldose isomerism, optical isomerism, and DL isomerism. In this session, we'll be discussing about epimerism. Objectives of the session are, at the end of the session, the learner shall be able to describe various aspects and significance of epimers and epimerism. The first thing is, what is epimerism? What are epimers? Two monosaccharides, okay, so differ in each other, differ in each other what? In their configuration, that is arrangements of the groups around carbon atom. In the configuration around how many carbons? Single carbon atom. Carbon atom. Two monosaccharides differ in each other in their configuration around single carbon atom. Those sugars are called epimers. Now I will explain by writing the structures. Let me write the structure. Okay, these are the structures A, B, and C. Suppose if I write this is glucose, all of these are glucose molecules. Okay, in this one what I do is, this is the second carbon, and this one is fourth carbon, and this is the fourth carbon. I will concentrate on this one. Okay, first let me compare A and B. Okay, what I do is now here, so first identify the second carbon in both the molecules. Okay, here what I do is I will change the configuration in the A. When I change the configuration in the A, let me see here. Okay, here what I do is like this. So here what happens here, by writing this configuration, in this one we have the molecule, around second carbon, the configuration you can compare between A and B. Okay, A and B. So in this condition what happens here, in the B, the OH hydroxyl group is on the right side, whereas the hydroxyl group is on the left side. Okay, the only difference between A and B is configuration around the second carbon. This is, we call it mannose. So mannose is an epimer of glucose. Okay, then let me see the C. See again at the fourth carbon, around this fourth carbon, here I am writing here, also around this carbon I am writing. Okay, let us see again. I am changing the configuration around the fourth carbon. Here and here it is. So around the fourth carbon there is a change in the configuration. The B and C, they differ in the configuration around the fourth carbon. So this one what we call is galactose. So galactose is an epimer of glucose. Mannose is an epimer of glucose. Galactose is an epimer of glucose. But mannose is not the epimer of galactose. Okay, then if you see the various carbons, C2, okay, C2 carbon, when you talk about the C2, what are the epimers? C2, what are the epimers? The epimers are glucose and mannose. And C4, the epimers are glucose and galactose. And C5, the epimers are glucose and L-idose. Okay, so these are the various aspects about the epimers. Okay, then let us talk about the significance. Second objective, significance. In this one, let us see one epimer. One epimer can be converted to another epimer. Epimer through process called epimerization. Through the process called epimerization. Okay. In this one, there are some enzymes are there which can convert one epimer to other epimer. So that is called epimerase. Okay. What is the significance of this one? Because if one epimer is available, other epimer we can get it. That means if glucose is available, glucose can be converted to galactose. Even galactose is not supplied, the cells can get the galactose from the glucose. So like that. Okay, this will study in the galactose metabolism, epimerase enzyme, role of the epimerase enzyme. So these are the various aspects and significance. Now I will summarize these aspects. Okay, so in this session we have taken up about the epimerism, objectives, various aspects and significance. Two monosaccharides differ in each other in their configuration around a single carbon atom. Okay, though they are called epimers. I have explained by taking up the glucose and where the configuration around the second carbon and fourth carbon. If there is a difference in the second carbon, that is mannose. If there is a difference in the fourth carbon, that is galactose. Mannose is an epimer of glucose. Galactose is an epimer of glucose. If C2, there is a difference, glucose and mannose, and C4, glucose and galactose, C5, glucose and L-idose. Significance, one epimer can be converted to other epimer. Okay, this process we call epimerization. The enzymes which play a role are epimerases. Okay, suppose in this condition, if glucose is available, galactose can be formed. If galactose is available, glucose can be formed. So this is about the epimers, epimerization and their significance. In the next session, we'll talk about the anonemers. Okay, thank you very much.